Morning everyone, thanks for joining this session. In the spirit of sharing something about myself, I grew up snorkeling on the beaches of Perth, Medham's Pool mainly, and then kind of thought I'd always be a wetland ecologist until I jumped in at Ningaloo Reef and saw corals for the first time and quickly got my diving ticket and moved to coral reef ecology. Also in the spirit of sharing something, I put in a very niche kind of abstract, but then was given a keynote, so my talk's quite different to what I initially thought I was doing. I'd also like to begin by acknowledging the traditional owners of the lands that we're meeting today, elders past, present and emerging, and particularly in the RRAP space as well, to all the traditional sea country that we work on. So to begin, I'm going to talk about one of the projects within EcoRAP specifically, and it's really kind of nailing, or trying to understand many of the gaps in the limitations to early coral recovery. We're a team between CSIRO and UQ, involving PhD students, postdocs and scientists, and we're about 22 people. Okay, so the integrity of coastal marine systems across the world has been degrading, and that's inclusive of coral reefs. These are many of the kind of reefs that we've been working in the last 3 years, and Roger fortunately has introduced many of the processes around resilience that forms the introduction of my talk. As we've all seen, we're currently in the middle of a mass coral bleaching event, and so once we get into this kind of state, what are the sorts of processes that can help recovery? So while ameliorating stresses of rest decline, boosting the resilience of coastal marine ecosystems relies extensively on ecological restoration. And that's one of our hopes, is that we can use restoration to help with resilience. So if we think about a hysteresis curve, we have positive and negative drivers, and they'll keep the system into states of, coral dominated versus not dominated states, and there are different components that can push us from or keep us within those positive states, and that's what we're aiming for. And overcoming recruitment limitation is one of those major drivers that can help keep a system in a state that has positive reinforcing drivers. So what do we know about coral recruitment ecology? We can break it down into stage-specific limitations that drive recovery, all the way beginning at reproduction to larval dispersal and then return back onto the reef, settlement and early growth and survival. And so we can break this down into stage-specific processes. Now we know that acute stresses really typically reduce fecundity of most corals on the reef, the survivors that is. So we have a lot less output, and what does get output is often not functioning very well. What do we know about fertilisation? Well, latent effects, they likely cause decreases in fertilisation, and that can be related to spacing of individuals, so that maybe the coral and sperm don't meet each other very well, but it also can be like, well, maybe those sperm aren't functioning very well for that year or a few years afterwards. We also know that chronic and latent effects definitely impact larval development and likely dispersal and reef connectivity as well. Now, predators and competitors influence larval supply and settlement, and likely the relationship between those. Post-settlement predation and competition are severe limitations to post-settlement survival. Now, once we're getting through to the larger size of recruits and then growing into mature adults, environmental gradients can influence the demographic rates that occur on reefs. But I would argue that across 4 of these processes, we actually have very limited understanding, and that's what ECO2 is being challenged with trying to address. We basically had no field attempts to validate alley effect theory. There's very few attempts to validate any connectivity models, and there's limited knowledge on presumed supply-settlement relationships, and I think there's no attempts to quantify the effect of environmental gradients on juvenile coral demographics, but there might be some that I'm not kind of aware of. So that's the overview of the talk. The beginning of the talk is we'll ask the questions, what are the critical densities of corals that are required to establish functional reproduction on a reef? And ask the question, can connectivity models capture larval supply at settlement that is at scales that are relevant to restoration operationally? And what are the taxon-specific sizes that may optimise early coral survival across environmental gradients to assist with densities of deployments on reefs? So this is going to be a little bit of a showcase because there's many people that have kind of contributed to this. So first off, Elizabeth Bucari is one of our PhD students, and she's been looking a lot at sperm limitation effects, and one of these is looking at the influence of wind on embryogenesis and larval survival. And she'd set up some pretty cool experiments using recirculating flumes driven by this wind turbulence that was downscaled from observations throughout the GBR at spawning times. This work was done at CSIM, and what she found is that fertilisation is inhibited by high wind and long exposures. She did this with 4 coral taxa. That also, well, as you increase this turbulence, we get more deformed, fragmented and damaged embryos. And what does that mean afterwards? Well, actually, larval survival is significantly higher for those intact embryos. So we understand from this piece of Liz's work that increased turbulence reduces the production of coral larvae by disrupting fertilisation and larval development. So we need to think about, well, what does that mean for outplanting in different kind of environmental settings? If you didn't see Pete's talk yesterday on our postdoc Jared Ricardo's work, there's a little snapshot about that. And this is where we ask, well, what are the adult patch characteristics required to have a functional reef in terms of reproduction? So for that work, we've conducted 5 manipulative field experiments manipulating coral densities out on reefs using small and low density patches. And what our field experiments showed, is that they resulted in low fertilisation success, demonstrating that alley effects do exist on a reef. So this has previously only been modelled. We then use this information plus a whole other bunch of parameters that have been collected in the lab, as well as some dispersal parameters out on the reef. And Jared put together this model. It was improved from the Teo and Todd model. And as you can see, we're getting this great match between our field validation experiment and what the model's producing. So what we can then do with this is we can ask the question, well, what are those colony sizes and spacings that we need to have some minimal kind of reproduction output? And, Jared's produced some really nice rules here that, we need partners within at least 5 metres of each other to have something meaningful for functional reproduction. And that kind of goes to about 9 average size colonies to achieve at least 10% reproduction. But, we want to get higher than that. So colonies can be spaced 2.5 metres apart, and they might be getting to 40% fertilisation, for example. So this is something that managers can use, or we can use in our decision making on outplanting on reefs. So Robert's been doing some work on ground truthing connectivity models, and we've done a series of experiments to understand, well, what's the demographic influx of coral larvae of spat onto reefs over different spawning events. So to do that, we've done this in the Cap Bunkers, but also in the Palm Island region. We set up these stations where we sample coral larvae in the plankton before and during spawning events every day spatially. We set up tiles as well, just to understand cumulative settlement just after that spawning period, just for a couple of weeks, so the very early settlement stage. Do some really high connectivity modelling. We're getting some really nice spatial patterns, which between these 3 events, the concentration of influx is agreeing between events. And what are we seeing in terms of our predictive ability? Well, when we look between model observations of settlement and what we observed, we're getting a significant prediction, but there is high variance around that. So there's some caution in what we can predict at such small scale. So we're talking about a kilometre here. But then when we only use our observational data and we match up supply with settlement, we see we get a significant relationship, and a lot of that variance is quite well explained as well. So we can use tiles to infer something about the supply of larvae is what this is suggesting. Now Devon's been looking at some work around size-based survival of juvenile coral outplants. So this is more looking at asexual fragmentation and rules that we can utilise. She's been deploying fragments at different kind of orientations and different species. This one's been going for about 15 months now, and she monitors that every 3 months. Before February, what we were seeing is between an acropora and platygyra is we're getting higher survival with the acropora, but that survival is decreasing with increasing size above 25 millimetres for these fragments, and there's no real effect of the orientation here. Now, in terms of bleaching incidents, the platygyra are bleaching regardless of their kind of size, but what we're seeing with the acroporids is actually a size escape. So anything smaller than 25 millimetres hasn't bleached, but then beyond that size, we're getting about a 50% bleaching incidence. So, and Devon's going back to herons soon to actually then do her mortality assessments. Now I'd just like to acknowledge the co-authors for this work, which is based on juvenile demographics across the Great Barrier Reef, and this fits in with the reference reef kind of design that we've utilised at EcoRAP. Hopefully you saw that in Katarina's talk yesterday. Our major objectives is to assess variation in juvenile coral demographics across coral groups and environmental gradients, and then to provide guidance on the sizes and densities of corals that maximise probabilities of survival following outplanting. And this knowledge can provide baseline counterfactual information and can also be utilised by the modellers. So how do we do that? Well, we've utilised the spatial design of EcoRAP, which goes from the far south to the far north and into the Torres Straits. We have nest sites and quadrats in a nested style design. And we've got 2 transitions because we've got 3 data points at this point in time. We only work in the 5-metre depth profile. So we define juvenile corals for this exercise as less than 40 millimetres. It's operational and it's kind of a classic size. And we're kind of using 5 major groups, and we're following about 2,500 individuals. How do we do it? Well, Katarina writes this on a data sheet underwater in T1, like T0 when we set it up, and someone has to transcribe that later. And then we actually take an image and we process that using Reef Cloud to get cover at that kind of scale, and also to give us an image here that we can work with, and then that gets traced onto a map, and we grid that up as well, so now we can look at density dependent things too. So this is in this kind of database, and then we go back to those plots, and you can start seeing things grow and die and appear, and then we create those mats, and then we can create our database from that. Some of the other data is about environmental parameters. Mariana's done all of this work, and you can see we've got a clear environmental gradient here with temperature going from south to north, and I think it's about 4 to 5 degrees there. And then we have other parameters that we can look at, and that's things such as turbidity, which is vertical attenuation, and you can see there's some areas of the reef that are more turbid than others, and the upwelling down south there's a clear signal of the chlorophyll A there. We can put this into an ordination, and when we do that, we end up with 3 pretty, I would call those quite distinct groups, where we've got this kind of central northern cluster. Is there a laser pointer? Do I dare? Up here, which is anything from the central reefs all the way to Torres Straits. Then we've got the Capricorn Bunkers out here, so they're the reefs in the far south, offshore, and then we've got the Keppel Islands, which are the reefs in the far south, but inshore. And the temperature gradients driving this grouping at the top, this separation, and then this is chlorophyll A, and this is turbidity. Okay, so I'm aware I don't have much time left, so I'm going to breeze through these. So just some data on coral juvenile density is that, unsurprisingly, acropora are the dominant corals in these fixed plots. throughout most of the reefs, apart from those in the very far north. And we do get some kind of community separation there, where the offshore northern reefs and the Torres Straits, in an ordination, do separate out. And if we're thinking about what densities we're looking for, well, total densities here, we're getting anything up to 26 juveniles per metre square, or as low as 15 juveniles per metre square. So in a healthy reef systems, which we've been working in the last 3 years, maybe these are the kind of densities that we need to be thinking about. We look at recruitment. We've got the highest in acropora, unsurprisingly, and when we look at mortality, we see the similar story. When we think about this thing called turnover, and it's just, well, what's the net gain or loss? You can see that as generally around 1 to 2 individuals per metre square per year. We've got a couple of hotspots there with, loads of acropora coming in still in the Capricorn Bunkers, and loads of porids coming in the Torres Straits. I think what's interesting is to think about, well, when we deploy, it might not be a single deployment event on a reef. We might have to do sequential deployments rather than just one event and assume that things are going to be cool thereafter. So some size-based data now. Unsurprisingly, this is growth. We get positive relationships with acropora and pocillopora. And then these kind of neutral or slightly negative size-based growth relationships with porids and merulinids. In terms of environmental parameters, there was no positive effects or negative effects of anything apart from flow, where we get a slight positive effect of flow on growth rates, which has been shown in other studies as well. We think about survival, we get this generic function, size-based survival. As things get bigger, they get smaller, and so the minimum size I think we're working there is about 3 millimetres, and it's going up to 40 millimetres, and we find that across corals and other benthic invertebrates. But if we think about what's happening across clusters, we get this thing that's happening at the Palm Islands, which is kind of not, which is this neutral kind of relationship where size is an increasing survival in our corals on the reef, but you can also see that palms have a higher general survival than all of the other reefs as well. The Keppels have this negative relationship, but that's data to porprait, so, and you can see there's huge variance around that, so I would take that with a grain of salt. Interesting, with temperature, we get positive and negative relationships. So we get a negative relationship with acropora survival as temperature increases. We get positive relationships with the merulinids and the porids, and then we get these temperature-independent relationships with the 2 other taxa. Remember, this is not any acute conditions. This is only that kind of chronic temperature gradient that is found naturally on the GBR. So what does this mean for restoration decisions? Well, size and early growth and their relationships with survival thresholds are really important to kind of keep in mind with outplanting, and that depends on where that is occurring. So, survival generally increases with size, but not necessarily for outplanted fragments, and there's, some small size escapes potentially, which are occurring with bleaching, which, is a general rule of thumb as well. And now we think about, well, how many colonies may we need on a reef? If we're doing outplanting and adults that are spawning, need to be closer than 5 metres to each other to have anything of some kind of functional reproduction of that reef. And so as things like wind and turbulence increase, we need those things coming closer together as well. So depending on the environmental setting, we have to think about different kinds of clustering. And finally, connectivity models. We need to make sure that when we're operational decision making, that scales need to match, and our trust in what they can do needs to be taken into consideration. And so, at larger scales, they're doing really well. As we get finer, they're sometimes doing well. And when we get to that really fine operational scale, we'll probably need blends between models and field data, unsurprisingly. I'd just like to acknowledge the EcoRAP leadership team, the EcoRAP 2 team, and like the massive EcoRAP team that we've been working with, the managing entity, GBRF, and the AIMS IP team, who have facilitated a lot of our free, prior and informed consent, all the stations we've been working with, and the Dargel Land and Sea Rangers, who we've had on all of our spawning events, and it's been a cool journey. Thank you, everyone.
Thank you, Chris. That was an absolutely perfectly orchestrated 20-minute talk. You had 5 seconds to go, so good job. Do we have any questions for Chris?
Thanks, Chris. Awesome work that you guys are doing. On the spacings of the reproductive colonies versus the occurrence of settlement, have you been looking at how those things correlate across the different coral families or genuses, or have you mostly been doing the acropora work for the adult spacings?
Yeah, right. So I think if I interpret your question, we could think about, well, in our juvenile demographic plots, what are the actual spacings in those relative to what kind of these rules about the functional reproduction? We haven't done that yet. This kind of analysis that I presented on the juvenile demographics is, I've kind of done that over the last month, and it's such a rich data set that I think that's a very worthwhile thing to do. I think also with the larger orthomosaics that we have in EcoRAP, and maybe Renata's going to talk about those later, a really obvious thing is to utilise those and to see, well, what are the spacings of species in those, and what does that mean related to our kind of reproduction alley effect modelling. One thing I will say with the juvenile plots is we can't get to species level. So you saw that there's broad groups, and if you've ever looked at a 1 centimetre acropora, they all look the same. If we track them for long enough, we might be able to get them into morphospecies, but I think it needs a geneticist to get it to our species. So, yeah.
Thanks, Chris. Great work. With regards to density-dependent fertilization, that's got real implications for rare species and has got real implications for the future of coral reefs as the densities are slowly declining. Do we need to start switching from thinking about density dependence to clumping?
To what, sorry?
Clumping. I mean, the distribution of species, especially with deployments. If we are deploying, we don't want to compromise one species over another by increasing the density of one species at the expense of others. Does deployment need to think switching to clustering colonies in clumps and bringing rare species together in order to give them a fighting chance to continue into the future? So it's not
that's
mean
a really
density that
question.
matters any longer, but it's the spatial distribution.
I think I'd ask first, why is the with a rare species, if it's naturally found spread apart, why is it rare? I think would be the first question. And do rare species kind of reproduce in what we understand with these really common acropora that we've been working with? Because our models have been parameterized from tenuis, acropora tenuis, acropora digitifera, and acropora hyacinthus. So really common, functionally kind of typical corals. But maybe the model would actually differ with a rare species. So I think we'd have to do some fundamental biology first, and then depending on that, we may have to then think about clumping much more and having less spread.
So if I can talk on that, I'm working in Kiribati where that acropora hyacinthus is extinct on many atolls, or it'd be one colony or two colonies. There's no reproduction going on. So what you're learning here is extremely relevant to the front line of collapse, where we must get those corals back together again, or there's no recovery. And so this is extremely relevant to what we're doing. That's what we're actually doing, is bringing them back together into clumps.
Yeah, so I think it's a safe option to have a high density clump, but you're also increasing your risk that if you have a disturbance and they're all in that one clump, and that disturbance hits that clump, you actually then have no resources outside of that clump. So, it's like a cost-benefit exercise in that sense as well. So, yeah.
Hi, thanks. Just picking up from that, it's also very relevant to the rear guard of collapse, so to speak. So I'm come here from Florida, and in Florida, you could argue that relatively speaking, all of our coral species are rare. now, but they weren't formerly rare. So your point about, why did they become rare? You could consider these to be formerly common species that are now becoming rare. And Florida's begun to do exactly what you've suggested, which is to clump corals together in what we call spawning hubs to try to increase the chance of them surviving. Unfortunately, there's these other effects that then hit those corals, for example, susceptibility to disease that is then hitting them because we put them together in these sort of highly dense aggregations. So, yeah, great talk, but definitely relevant to lots of different places around the world. I guess that's not really a question.
Time for one more question.
Nice comment.
Hi, thanks. Bit of a change in direction, but just with the modelling, so presumably where you find better survival of recruits, better survival of juveniles, you're going to have better recruitment anyway. So if you're planning an intervention and you've got this thing of the best place to plant is where presumably the best place natural recruitment has already got the best chance, how do the models fit with like how you plan an intervention based on you really want to help areas that aren't helping themselves?
Sorry, so can you repeat your question?
Yeah. No.
You can't remember it either.
It's basically, so we do a lot of coral planting. We've done it since 2018, but the issue is where it's most effective to spend limited resources. So trying not to plant corals where the natural recruitment is occurring anyway, and they seem to line up. The places where you're going to get best survivorship is where the substrate conditions best suit natural recovery anyway, and you may not be producing a big environmental benefit. So how does the modelling, say, help pick sites, and how do you factor in that consideration of best utilising your available resources?
Yeah, I think I'd ask the question first, well, why aren't those areas naturally recovering, whereas others have all of this natural recruitment there? And if it's something like, if it's a predator that's killing everything in those areas where corals aren't naturally recruiting.
This isn't a time scale where you haven't actually figured out. Like say you have an impact. Sorry, this is within a certain time scale. Like say, for example, now you've got an impact. You wait a certain amount of time before you decide whether to intervene or not. But you really want to be intervening at areas where there's not enough natural recovery. But how can you figure out, like, how long do you have to wait? So far, we've waited, say, 8 years since 2016. We've got sites that are 200 meters apart. One's recovered really well, the others hasn't recovered very well at all. How do you actually use this information about juvenile survival to help decide what to do and where to do it? You want me to be more concise?
Yeah, no. I think, I mean, it might not, like the juvenile survival, we need to understand why they're not surviving there. Is it competition? Is it sediment smothering? Is it predation? So we need to kind of understand, or is it something environmental? Is it, super bad water quality? Although you did say it's 200 meters apart, so I doubt it. So there might be something some ecological interaction that's occurring there that might be restricting the survival of those colonies. But I think without diagnosing that, and if you only use, say, a size escape rule, well, if you outplant bigger ones, they might survive better. But if there's a lot of corallivellas there, they're probably not going to survive very well if you put out bigger ones, because they're like pop sticks out in a desert. And that's when you would be better putting out smaller ones so they have a chance to kind of establish themselves and grow before they get hit by a predator. On the other hand, if there's a lot of competition and you put out a small coral, they might get overgrown quickly. So it's kind of a trade-off, and we need to understand the strengths of these ecological interactions to really understand, to really understand what the best strategy is going to be. Sorry, I mean, that's probably not a very satisfying question, but I don't think it's a simple problem.
I might talk to you afterwards as well, if possible.
No worries.
Thanks.
Sorry. I think that's all we've got time for, so thank you very much, Chris.